My name is Leslie Castillo, and this is my answer to question number 1. PCR, polymerase chain reaction, is a method that is used to make billions of copies of a DNA sample. The purpose is to copy this DNA and make billions of copies in the vitro way, meaning that it is in the test tube or outside of the body. There are 3 steps for this method. The first step is denaturation. This is when the DNA is heated up to a semi-high temperature, and this is done so in order to separate the hydrogen bonds that are connecting the double-stranded DNA molecule. The temperature that it rises up to could be possibly like 94 degrees Celsius. Step 2 is priming. DNA primers, which are short DNA fragments, which are made up of DNA that are binded to your DNA sequence, are included in the priming step. Also, DNA polymerase is present as well to continue replicating the daughter strand and free DNA nucleotides to build the new daughter strands. DNA nucleotides such as the ACTG. However, the DNA polymerase that is being used is a special kind, and it is called the TAQ, spelled TAQ, DNA polymerase. And the reason being that is that you cannot use normal DNA polymerase due to the fact that you are using high temperatures during this method, and it would not survive. Instead, by using the TAQ DNA polymerase, it is not heat sensitive, so it would be able to survive during this method. And also during the priming stage, you would cool down the DNA, bringing it down to approximately a 65 degree Celsius temperature so that the double-stranded molecules will form. The third step would be extension, which would be where the temperature is raised slightly in order to increase the rate of replication through DNA polymerase. One thing to mention is that during this method, it is done so in the labs, and they use a machine called the thermocycler. Also, when DNA is going through the PCR method, it increases the number of DNA you have exponentially, meaning that during the first trial, you'll start off with 1 DNA sequence, and then from there it will duplicate to 4, and then from 4 it would go to 8. My name is Leslie Castillo, and this is my answer to question number 2. So basically, viruses are extremely small in size. They aren't cells. They are infectious. They do infect humans, animals, plants, and even bacteria. When the virus enters the host cell, basically what happens is they take over the host cell's enzymes to replicate inside of the host cells. Viroids are extremely small in size as well. However, they are smaller than viruses. They are circular-shaped pieces of short-stranded RNA. They only cause disease in plants that is known of. What happens is that the viroid RNA attaches to plant RNA to prevent translation of plant RNA. This causes the double-stranded RNA to be degraded, which means the plant doesn't produce protein, which leads to the disease. And prions, what they are is they are small in size, and they infect animals. They are a PRP. Basically, prion PRP causes a normal protein, which is a cellular PRP protein, to refold into prion PRP. So it goes from beta helices to beta sheets. So basically what happens is that the cellular PRP protein refolds into prion PRP, which is the disease causing protein. How they are similar is that they are all infectious and can cause disease. How they are unique is that they all infect their own designated organisms. Now, comparing bacteria to viruses, prions, and viroids, they are not the same. Viruses are non-living. They are not made up of cells. They are small in size. They cannot reproduce outside of the host cell. Whereas bacteria is alive. They are single-celled organisms. They are large in size, and they can reproduce and grow.
My name is Leslie Castillo, and this is the answer to question number 3. The first method would be boiling. This is a moist heat category, and it is used to disinfect, sanitize, sterilize, and pasteurize. The way that it works is that it denatures proteins and destroys cell membranes. As we all know, water boils at 100 degrees Celsius. However, this is at sea level. Another thing to mention is that depending on the elevations, so for example, we're at sea level, so water boils at 100 degrees Celsius, but if it were at a different elevation, it would require a different temperature. The second one would be autoclave. This is also a moist heat category, and this is when pressure is applied to the boiling water. When this happens, it allows the boiling point temperature to rise. Therefore, it would be hotter than if you were to boil water. And the third method would be filtration, and this is the passage of fluid through a sieve designed to catch microbes. And sieve is another word for filter. Here I have demonstrated what a filtration process looks like. So at the top there is the non-sterile medium, in the middle there is a filter, and at the bottom would be the sterile medium. There are different types of filters with different pore sizes. It can range anything from 0.01 to 5. One of the most common filters is the high efficiency particulate air filters. It is most common because it is 99.97% effective in terms of filtration. And to answer the second part of this question, boiling and autoclave endospores would not be able to survive. It would not be an effective method due to the fact that endospores are able to survive high temperatures, and that is what these two methods require, are high temperatures. However, in the third one, endospores would not be able to survive due to the fact that if they use a certain size filter, the filter would be able to catch the endospores and it would not be distributed into the sterile medium. For example, if you were to use the HEPA filter, which is the high efficiency particulate air filter, you would be able to catch those endospores.
My name is Leslie Castillo, and this is the answer to question number 4. For A, it is penicillin. Penicillin is a part of the beta-lactam group, which prevents cross-linkage of NAM. How it works is that over time with penicillin, the bacterial wells start to weaken and eventually break open. The only thing is that it only affects growing cells, not cells that have been produced and dormant. For B, tetracycline. Tetracycline blocks the A site of the ribosome. It is located on the right side of the ribosome. And what it happens is that tetracycline does not allow the new transfer of RNA in carrying amino acids. So basically, it stops the production of new amino acids to be let in. For C, it's amphotericin B, and this prevents proper cell membrane formation. However, this medication only works for fungal infections. It does not work for bacterial infections. And the way that it works is that in a fungal cell membrane, there's a steroid molecule named ergosterol. And whenever this steroid molecule is present in the fungal membrane, then this medication jumps in and creates a hole in the membrane to prevent the cell membrane to actually form completely. And for D, it's sulfonamide. Basically, this prevents or inhibits folic acid synthesis in pathogens only. What it does is it blocks the binding site of para-aminobenzoic acid, and bacterial cells cannot produce DNA and RNA. What this means is that it basically cannot divide.
My name is Leslie Castillo, and this is my answer to question number 5. Virulence factors are the factors that contribute to virulence, which is the degree of pathogenicity, and that is how easy it is for the pathogen to spread and cause disease throughout the host. One thing about it is that it's unable to tell the severity of the disease. So 4 virulence factors. The first one is extracellular enzymes. These enzymes are secreted by pathogens that contribute to invade the body and avoid the body defenses. Many of these pathogens have mutant versions which don't make the enzymes and don't cause disease. The second one is exotoxins, which is a type of toxin, and toxins are chemicals that harm the host. There are 2 groups, which are the ones that I'm going to mention. So exotoxins are secreted by live microbes. They destroy the cell or interfere with normal metabolic reactions. A way to protect from toxins from exotoxins is to produce antibodies and toxoids. Third is endotoxins, and they come from lipid A gram-negative cells only. They are found on the lipopolysaccharide membrane layer, and it is only released once the bacterium is dead. The fourth one is antiphagocytic factors, and these factors, they are factors that prevent phagocytosis by white blood cells, and it includes the microbes with bacterial capsules and antiphagocytic chemicals. Now, normal flora, it can produce virulence factors. Normal flora is a mixture of organisms that live in humans that are healthy. Basically, when the microbes attach to the host cell is when it would be able to produce the virulence factors.